Welcome, everyone. I'm excited to introduce today's topic, studying vegetation. On this first slide, we see that the community is a larger unit than just the population. It can range from a tiny patch of land or water to vast forests. Over the course of this presentation, we'll explore how vegetation communities are defined, their significance, and how they vary in size. Let's dive in and discover the fascinating world of vegetation together. Let's take a closer look at the different types of communities. First, we have minor communities. These are communities that are heavily influenced by the neighboring communities around them. Their development and behavior tend to be shaped by the inputs and interactions with adjacent groups. On the other hand, we have major communities. These are more independent and self-sufficient. They don't rely as much on their surroundings for resources or influence. Instead, they tend to operate more autonomously within their habitat. Understanding these distinctions is important because it helps us see how community dynamics function differently based on their level of dependence and independence. This concept ties into the overall theme of how communities interact within their environments and influence each other. Today, we're going to look at the methods used to study vegetation. There are two main approaches that researchers typically use. The first is called the plot methods. These involve setting up specific plots or plots of land where detailed measurements and observations are made. This approach allows for precise data collection about the vegetation within a defined area. The second method is known as plotless methods. Unlike the plot methods, these do not require establishing specific plots. Instead, they focus on sampling techniques that can quickly estimate vegetation characteristics over a larger area without the need for designated plots. Both methods have their advantages and are chosen based on the specific goals of the study, the type of vegetation, and the resources available. Understanding these methods helps us better interpret how scientists gather data about plant communities and their environments. Today, I want to talk about the plot methods we use, starting with the concept of a frame. A frame, in this context, is a structure that has a fixed shape and area, meaning it doesn't change size or form during measurement. This stability is important because it provides a consistent reference point for our measurements. Now, when we think about the shapes of these frames, there are a few common types. First, we have the square, which is simple and easy to work with because all sides are equal and angles are right angles. Next, there's the rectangular shape, which is similar to the square but with different lengths for the sides, so it's longer in one dimension than the other. And finally, we have the circular shape, which is quite different from the squares and rectangles. Circles are unique because they have no corners or edges, and their curvature makes them useful in many specific measurement scenarios. Overall, choosing the right shape for your frame depends on what you're measuring and the context of your project. These shape options give us flexibility and accuracy in plotting our data or conducting measurements effectively. So let's talk about what a quadrat actually is. Essentially, a quadrat is a defined area with a specific shape and size that we use for sampling in ecological studies. It helps us focus on a particular patch of habitat so we can study the organisms within it systematically. Now, according to BCFT in 1953, a quadrat can be described as a small field study unit or sample area, often with a square meter or sometimes 1,000 square meters, what we call a milliacre. The idea here is to establish a manageable, well-defined zone where detailed observations can be made. Similarly, SAF in 1983 emphasizes that a quadrat is a small, clearly demarcated sample area of known size. This clarity in boundaries is crucial because it ensures that the data we collect is precise and repeatable. Throughout our presentation, you'll notice how important it is to have a standardized area for sampling. It allows us to compare data across different studies and habitats reliably. So, in essence, a quadrat is a fundamental tool in ecology that helps us quantify and understand the distribution and abundance of organisms in a specific, manageable area. Now, let's talk about the different kinds of quadrats. When we list quadrats, we're essentially marking out simple sampling areas of a predetermined size. Once we've set these areas, we carefully record the names of all the species that occur within each quadrat. This method helps us gather detailed information about the composition of the community within that specific area. One of the main uses of this approach is to study the degree of dispersion, or how frequently different species occur across the area. By doing this, we can understand patterns of distribution and how species are spread out in their environment. Overall, quadrats are a fundamental tool in ecology because they give us a snapshot of species presence and abundance, helping us analyze ecological relationships and community structure in a systematic way. Now, let's talk about count quadrats, which are also known as list count quadrats. These are a specific type of quadrat used in ecological studies. When we use count quadrats, we're not just noting which species are present, but also counting the number of individuals for each species within the quadrat. This allows us to gather important data on how abundant each species is and how densely they are distributed across an area. By doing this, we can get a clearer picture of the population sizes and the relative abundance of different species in the ecosystem. So, essentially, count quadrats help us quantify and analyze the abundance and density of species, which is crucial for understanding ecological dynamics and making informed conservation decisions. Let's talk about chart quadrats. When we want to study species distribution in a way that's true to scale, we need a method to record individual organisms accurately. That's where the chart quadrat comes in. Essentially, we use a miniature quadrat on graph paper, which is scaled down appropriately to match the actual size of the area we're studying. This allows us to document precisely where each organism is located within the quadrat. The key idea here is that by doing this on a graph paper, we can analyze the spatial distribution more easily and accurately. This method is particularly useful because it gives us a clear visual record that can be scaled back up if needed. Overall, chart quadrats are a valuable tool for detailed, spatially accurate studies of species distribution, helping us understand how organisms are arranged within their environment. Let's talk about the permanent quadrat, also known as the fixed quadrat. This method involves leaving the quadrat undisturbed after we've charted the vegetation within it. The key idea here is that we don't move or change the quadrat once it's set up. Instead, we revisit the same area, same site, over a long period of time. This allows us to observe how the vegetation changes or remains stable over time, providing valuable data on ecological succession, growth patterns, or environmental impacts. The main point to remember is that the permanent quadrat is all about consistency, studying the same spot repeatedly to understand long-term ecological processes. It ties into our overall theme of monitoring changes in ecosystems and understanding how environments evolve. The size of a quadrat varies depending on the type of vegetation you want to study. For small plants like mosses, lichens, and liveworts that grow in patches, we typically use small quadrats, around 20 centimeters by 20 centimeters. When we're working in grasslands that are of a uniform or pure type, a larger quadrat of about 50 centimeters by 50 centimeters is more appropriate. And in areas with a high diversity of plant species, we need even larger quadrats, roughly 1 meter by 1 meter, to get a representative sample. It's also important to consider how many quadrats we need to sample. Generally, we aim to cover about 5 to 10% of the total area under study. This helps ensure our sampling is representative without being overly exhaustive. Finally, there's a minimum size for quadrats to ensure accuracy and consistency in our data collection. So, choosing the right size and number of quadrats is crucial for reliable results in ecological studies. Let's talk about plotless methods, which are also known as point methods. These involve sampling at specific points on the ground rather than using a fixed plot. The idea is to select several points randomly or systematically across the study area to gather data. Now, these methods are further divided into two main types. The first is the point frame method, where points are chosen based on a grid or frame arranged over the area. The second is the point-centered quarter method, which involves measuring distances from a central point to the nearest trees or features in each quarter around that point. Both approaches are useful, because they allow us to efficiently estimate things like tree density or distribution without the need for a defined plot boundary. They're especially handy in irregular or difficult terrains where traditional plot methods might be challenging to implement. So, in essence, these point methods provide flexible and effective ways to gather spatial data, helping us understand the landscape or forest structure more comprehensively. Now, let's talk about the point frame method, which was described by Levy and Madden way back in 1933. The core idea here is to use a scale-like frame that typically has about 10 holes. Think of it like a small grid or sieve that can be easily handled in the field. The frame itself is designed with ends that are fitted onto a pair of sturdy legs, making it portable and easy to position. One important detail is the length of the nails used with this frame. They are kept at around 60 centimeters. This length helps ensure stability and consistency when you're placing the frame at different points in the field. Speaking of placement, the key is to position this frame at several different locations across the field. This allows for a representative sampling, giving you a more accurate picture of the overall conditions or characteristics you're studying. Overall, the point frame method is a practical, systematic way to sample and analyze various field attributes, and it connects to our broader theme of reliable, standardized field measurement techniques. Now, let's look at the point-scented quarter method. This technique was proposed by Cottam and Curtis back in 1956, and one of its main advantages is that it avoids using a traditional frame, which can sometimes be restrictive. In practice, you start by fitting a brass needle or a nail on top of a rubber cork. This setup allows the needle to rotate freely, which is essential for the randomization process. Next, you fix the compass needle at a random position or in the direction you desire, depending on your study's needs. This randomness is crucial because it helps eliminate bias in your sampling. Then, you divide the area around the needle into 4 equal parts or quarters. These quarters serve as the sampling points, and by doing this, you effectively simplify the process of locating points within the study area. Overall, this method provides a straightforward, unbiased way to sample points in the field, especially useful in ecology and environmental studies, where avoiding frame bias is important. It's a clever technique that leverages simple tools to ensure randomness and accuracy in your sampling process. Let's take a closer look at how we quantify the presence of different species in our study area. First, we have the number of points with a particular species. To find its relative frequency, we divide that number by the total number of points surveyed, then multiply by 100 to express it as a percentage. This tells us how common a species is relative to the entire survey. Moving on, we consider the distribution of all recorded species. To estimate the average distance between points, we take the total distance of all species recorded and divide it by 4 times the number of points. This gives us an idea of how spread out the species are within the area. From this, we can calculate the average area occupied by each species by multiplying the average distance by 2, and then expressing that as a percentage of the total area, which we consider as 100%. This helps us understand the spatial extent of each species. Next, we look at absolute species density, which involves dividing the total number of individuals of all species recorded across all points by the total area in square centimeters. This provides a raw count of how densely populated the area is with each species. Similarly, relative density is calculated by dividing the number of individuals of a species at all points by 4 times the number of points, then multiplying by 100. This percentage indicates how dominant or common a species is in relation to the entire survey. Finally, we determine the absolute density of a species by dividing its total number of individuals by 100. This standardizes the density measure, making it easier to compare across different species or studies. Throughout this process, the goal is to understand not just how many species are present, but also how they are distributed and how dense their populations are within the area. These calculations help us paint a comprehensive picture of the ecological dynamics at play.